Okay, so we are going to talk about the Benedict's test to test for reducing sugars. Let's just start with how we do the test for the reducing sugar itself. So if we start off with 2 tubes, okay, and in each we will add a solution to test for. So in one, I will add some glucose, which is a reducing sugar. And in the other, I will add some water. Okay, and to both I will add the same volume of Benedict's reagent. And we'll now put this in the water bath around 80 degrees C for about 5 minutes. And we are back. There we are. So if I can just bring that close up. After about 5 minutes, you can see that when the glucose was present, a nice brick red precipitate has formed, and where we just used water, there has been no color change at all. Okay, so that's the straightforward Benedict's test to check for the presence of a reducing sugar. However, what if you have a solution whose concentration is unknown? And so the next part of the video we're going to discuss how to determine the concentration, or how to quantify reducing sugars. Okay, so this is useful where if you want to check the progress of a reaction, so you can take solutions out at different times and then check for how much reducing sugar is there. Or if you have one unknown solution and you just want to check how much reducing sugar is in that solution, or its concentration. So how do we go about it? So here we have the unknown solution, as you can see with the question mark right there. And I don't know the concentration of that solution. Okay, so how do I go about determining the concentration? What I need is a series of solutions of known concentrations. Okay, so I have 1%, got 0.5%, okay, got 0.1%, 0.25%, and 0.05%. Okay, so a range of solutions, all with reducing sugar in them and with known concentrations. And what I do then is just repeat the Benedict's test for all of those solutions. Okay, so add known amounts of each. To each tube. And then to all of my standard solutions, I add the same volume of Benedict's reagent. Once that's done, we also repeat the test for our unknown solution in the same way. So we take the same volume of unknown test solution, the same volume of Benedict's reagent, and then again to start the reaction we have to put it in the water bath, again 80 degrees Celsius for 5 minutes should do the trick. So after about 5 minutes in the water bath, here we have the results of our experiment. As you can see, depending on the amount or the concentration of reducing sugar in our standard solutions, the reaction has gone further to completion. So the more reducing sugar you have, the more the copper has reacted to form the copper oxide, and therefore the more color change you get as the percentage increases from 0.05% to 1%. Okay, now a very easy but still qualitative way to estimate the amount of reducing sugar in our test solution would be a simple comparison which of these tubes is the color is closest to. So I would estimate that it's around about between 0.05 and 0.1%, okay, just in terms of simple color comparison. But that is not quantitative, as I said. That's still a subjective way to estimate that concentration. It's still depending on the observer's point of view of what color it's closest to. So a more accurate way of doing this would then be to make what's called a calibration curve of the absorbances of these solutions. So actually measuring quantitatively the amount of reaction that's occurred in all these tubes. And I'll show you next how to do that. Okay, so what we are going to do is then to measure the amount of copper ions left in solution. Essentially, that depends on absorbance or transmission of light of particular wavelengths. Now, what you can't do with these solutions is to measure absorbance or transmission, because they are all different colors. And for colorimetry, you need to be measuring the transmission or absorbance of a particular color, i.e. a particular wavelength of light. So what we're going to do is to filter these solutions, remove the precipitate that's formed, and what we should have is a series of solutions which are all the same color. So what I've done now is just to filter away all the precipitate. So I poured the solutions through a filter paper and into a second test tube. So that removes all the red precipitate that's formed, and what's left is, in theory, just the remaining copper ions in solution. So we're just looking now at a more uniform set of solutions in which the only difference is the amount of copper that's left in the solution. And what you should see is that as the concentration of the glucose got higher and higher, more and more of the copper reacted in the standard set of solutions. This one is our test solution, and we're going to compare the color of this solution to the others in our once we do the colorimetry. So what we then do is to use a colorimeter and measure the absorbance or the transmission of light through that solution. Now what you want to use is a red filter as the solution is blue. Okay, right. Okay, so if we now use a colorimeter and measure the absorbance of the different solutions, we would then make a calibration curve using first our known concentrations of glucose. So for every concentration of glucose, I would get a particular absorbance. So the lower the percentage of glucose, I would get a higher absorbance because there's more unreacted copper in there. And then as the percentage of glucose would increase, that absorbance would go down. Okay, I'll get something like that. Right. Then I could make a curve through that graph. And that would be my calibration curve for my standard set of solutions. And then all I'd have to do to measure the concentration in my unknown solution is to also run that through the colorimeter, measure the absorbance, and I might get an absorbance around about there, for example, just for example. Okay, so if it was there, if my absorbance was there, then I just use the calibration curve with my known solutions to then determine the amount in the unknown solution. So I would do an interpolation, something like this, reading along the line. Wherever it hits the line, I then read downwards. And that would be my percentage of glucose in the unknown solution. I think that's about that. So remember, you can use this kind of quantitative test with pretty much any kind of reaction that shows a color change. You can use it to measure the progress of the reaction. As more of the product is formed, you're going to get a change in color at different time points. Or you could, as I've done today, you could just use it to test an unknown solution against a series of known solutions from which you make a calibration curve. Good luck.